Okay, now let's go to pharmacogenetics. So pharmacogenetics is a study of genetically controlled variations in drug response. Pharmacogenetics is the special area of biochemical genetics that deals with variation in drug response and the contribution of genetics to such variation. It is also a subset of pharmacogenomics and is the study of variations in DNA sequence as related to drug response. So there are a few key concepts or terms you have to understand and memorize. So the first one is monogenic. Monogenic is a genetic variation due to allelic variation at a single gene. Okay, so what does it mean by this? For example, gene of eye color. All right, so you have a brown eye allele and also a blue eye allele, but because brown eye allele is the dominant one, so the phenotype that appears in your eye color is brown. So that's called monogenic. It is due to the allelic variation at a single gene. So only one gene, but there are two types of alleles involved. For example, the brown and blue eye color. And then the second one is polygenic. It is due to variations at two or more genes, right? So, for example, the hair color and then combination with eye color. So, you have variations at two or more genes. And then the third one is polymorphic. It is frequently occurring monogenic variants occurring at a frequency of more than 1%. So, frequently occurring monogenic variants. It is monogenic variants, but because it occurs in more than 1% of the population, then we call it as polymorphic or polymorphism. For example, in a population of 100 people, and then you have 20% blue eye, and then 30% green eyes, and then 50% brown eyes. So we call this as polymorphic because the population that has blue eyes is 20%, which is more than 1%. And then green eyes 30%, which is also more than 1%. So it's called as polymorphic. So this graph shows you the normally distributed variation. So on the x-axis, you have the activity. Okay, this is, for example, the activity of an enzyme. Let's take an enzyme of cytochrome. So most of the population actually have this much of activity. Okay, the frequency here means the number of people, number of sample or human lah. Okay, so in a normally distributed population, the majority of the people in that population has this much amount of activity. Only small portion have very high activity or very low activity. But in polymorphic distribution, you can see that whereas the majority of the population have this amount of activity, but there are also quite many samples also have this lower activity. So because of this, so this amount represents more than 1% of the population, then that's why it's known as polymorphic distribution. All right. All right, so these two pie charts are showing you the genetic polymorphisms in drug metabolizing enzymes. So recall back your pharmacology. We have the pharmacokinetics, ADME, the absorption, distribution, metabolism, or biotransformation. And the last one is excretion. So the third one is metabolism or biotransformation. So remember again, in metabolism or biotransformation, there are two phases, phase 1 and phase 2, primarily happening in the liver. Okay, so in the phase 1, if you recall again, there are three chemical reactions involved: the oxidation, reduction, and also hydrolysis. Okay, oxidation, reduction, and hydrolysis. And then phase 2 is the conjugation. Conjugation means the addition of small molecules to the metabolites from phase 1 or the parent compound itself. Because when the drug comes into our body, it can undergo phase 1 and then phase 2, but it can also go phase 1 only and then directly eliminated or excreted. But there are also drugs that goes to phase 2 directly, and there are also that that does not go any metabolism. It is excreted directly as a parent compound. So the conjugation can be in the form of glucuronide, and then glycine, ethyl, methylation, and so on and so forth. So in the phase 1, for these three chemical reactions, oxidation, reduction, and hydrolysis, most of these chemical reactions are governed or are carried out by cytochrome. Okay, so cytochrome is the main enzyme in the liver that works to metabolize all the drugs that come into the body. So if you see the pie chart here, cytochrome P3A4 and 5 and 7. So remember, there are many species of cytochrome P450, and there are many families. If I'm not mistaken, there are 17 families, and then there are also subfamilies. So these are the cytochrome enzymes that have the most genetic polymorphisms: CYP3A4, 5, 6, 7, followed by CYP2D6 and CYP2C9. And in phase 2, it is not so. Phase 2 conjugation of the metabolites are primarily carried out by transferase enzyme. Okay, it's no longer cytochrome P450. So the most enzyme that has genetic polymorphism is UGT, which is UDP glucuronyl transferase. Glucosyl transferase, if I'm not mistaken, and then ST is sulfotransferase. So these are the two transferase enzymes in phase 2 metabolism that have the most genetic polymorphisms. Okay, so if there is a variation in this enzyme, for example here CYP2C9, so this enzyme is known to metabolize warfarin. Warfarin is a drug to thin the blood, to make it less viscous. So if there is variation in this enzyme, it either can reduce or increase the activity of CYP2C9. So what happens if it reduces the activity of CYP2C9? Then that means it has a reduced capability to metabolize warfarin. And when it has reduced capability of metabolizing warfarin, then you will have more active warfarin in the blood circulation, and this will cause overdose, and this will cause side effects. And if CYP2C9 is increased, then you will have less warfarin, active warfarin in the blood circulation. Then you will not reach the pharmacological effect that you desire. Alright, so this table is showing you the examples of genetic polymorphisms of drug metabolizing enzymes and drug targets for which genetic tests may affect drug choice. So if you have a prior genetic test before prescribing any medication to the patients, then you would know if the patient has a variation in certain metabolizing enzymes, and that may affect how you decide the prescription, what kind of drugs you want to give to the patients for. And So this genetic test can also affect the dose and reduce adverse reactions. Okay, maybe because it has a reduced capability of metabolizing that particular drug that you want to prescribe, then you would lower down the dose. Okay. Okay. The first example is NET2. NET2 is N-acetyltransferase 2. It is so this enzyme, this metabolizing enzyme, it targets isoniazid, which is an antibiotic. So what happens if you give the same amount or the same dosage of isoniazid to this variant person, then of course it will result in overdose, because it cannot metabolize isoniazid perfectly, and thus it has a higher concentration of active drug circulating in the system, and that can cause hepatotoxicity. And then CYP to C9, this is the example that I mentioned earlier. It metabolizes warfarin and acenocumarole. So in the case of it has reduced capability of metabolizing warfarin, then it will cause hemorrhage, intestinal hemorrhage, because the blood is too thin. And then CYP2D6, it targets the antipsychotic drugs and antidepressants. So the variation in the enzyme can cause extrapyramidal symptoms, cardiotoxicity, adverse drug reactions in poor metabolizers, lack of response in ultra-rapid metabolizers. And the interesting one is codeine. Codeine is a prodrug, if you still remember. Prodrug is a drug that is administered into the human body in inactive form. So it's not active when it enters the body. Then it has to undergo metabolism. Then when it undergoes metabolism, then it becomes active. So if there is a variation in this enzyme, and this enzyme cannot metabolize or has reduced capability to metabolize codeine, then you will have less active codeine. And the function of codeine is a painkiller. So when you have less active codeine, then you will have a lack of analgesic effect. All right. Then you can read the rest of the table by yourself. There are three mechanisms on how genetic influences pharmacotherapy. Okay, three mechanisms on how genetic influences pharmacotherapy. So the first one is genetic polymorphism of genes, which results in altered metabolism of drugs. For example, metabolism of tricyclic antidepressant. So the altered metabolism can result in increased or decreased metabolism of a drug, may change its concentration of active, inactive, or toxic metabolite. So this is what I have mentioned earlier. So this is like a wrap up. The increase or increased metabolism can result in change of active, inactive, and toxic metabolites. The second mechanism is genetic variations may produce unexpected drug effect or toxicity, or it may be lethal to patient. For example, G6PD deficiency or glucose 6-phosphate dehydrogenase deficiency. So this is a genetic variance that happens in certain human beings, and it is so the function of G6PD is to retain or to protect the integrity of red blood cells. So if you have a deficiency in G6PD, then it cannot protect the red blood cells, and that will result in hemolysis, the breakdown of red blood cells. And then the third one is genetic variation in drug targets. Okay, just now it's the metabolizing enzymes, and this is also protein that protects, that maintains the function of our cellular functions. And the third one is genetic variation in drug targets. For example, this may alter the clinical response. Okay, for example, you have a drug You have a drug A that targets a receptor, for example, receptor beta adrenergic receptor. Okay, so drug A's function is to inhibit beta adrenergic receptor. However, because it has a genetic variation, then the binding site of the beta adrenergic receptor is different. Okay, so when it is different, the drug A cannot bind to beta adrenergic receptor, and it can no longer inhibit the effect of beta adrenergic receptor, and that will alter the clinical response that you expect. You were expecting by giving or administering the inhibitor of beta adrenergic receptor, then you would have reduced activity. But in the genetic variation people, this does not happen. Then it just happens like normally happen. And then it also may alter the frequency of side effects. And then because of the genetic variation and the change in the binding site, instead of binding to the drug that it is supposed to be binding, however, it may bind to other types of drugs. For example, it binds to beta agonist instead of inhibitor. It binds to the beta agonist that potentiates the response. So variants of beta adrenergic receptor alter response to beta agonist in asthmatic patients.